Hi, and welcome to this revision mind map lesson. We're going to run through everything relating to module 5.3, neuronal communication from the OCR A-level biology syllabus. Now, 5.3 actually kind of fits nicely with a bit of 5.5. So actually this is going to be all of 5.3, and also it's going to be the animal responses stuff from module 5.5. And we should be able to get it all on this mind map. So first thing to do is you need to print this page out in A3 if possible. I will just disappear for a second if you would like to take a screen grab. Or you could print it out in A4. Again, I'll disappear for a second. You could take a screen grab. And then you could cut out these pictures and use it to create your mind map. Eventually, it's going to look a little bit like this, but actually we're going to do it again and I'm going to do it better than this. Okay, so let's get going. Okay, we're going to start in the center. We're going to zoom right in. We're going to talk about how the nervous system is arranged in humans. So the nervous system can be split into two things. First of all, we've got the CNS. Okay, now this stands for central nervous system. So if you have space, you could write central nervous system there. And then we've also got the peripheral nervous system. So the peripheral nervous system is everything that is not the central nervous system. Now the central nervous system is the brain and the spinal cord. And everything else is peripheral. So we're going to talk about the brain in a minute and the sort of structure of it over here. Okay, here we can see the brain. We're going to talk about the different parts of the brain and what they all do, but yeah, we're going to talk about that in a second. So in the peripheral nervous system, we can further divide it up into both the sensory and the motor nervous system. So sensory is this one, and the motor is this one. So a little bit of a subnote on the sensory. So this carries impulses and information to the CNS. So for example, two parts of the sensory nervous system which you might have heard about, maybe the optic nerve, you know about that potentially. That's the nerve that travels from the retina to the brain. That would be part of the sensory nervous system. And in fact, any nerve that carries sensory information to the brain, I think the one from the ear, I think, is called the auditory nerve. It's another one if you wanted to write that down. So the motor nervous system can be further subdivided into the autonomic and the somatic system. So the somatic goes here, and here is going to be the autonomic. So the autonomic nervous system kind of means, you know, we can put a little subnote to the side. It's a bit like the automatic. Okay, so it's not under conscious control. So you can't decide to, you know, I'm going to fire part of my autonomic nervous system just by thinking about it. It happens subconsciously. And we can divide it up actually into two key areas, or two key branches. And we've got the sympathetic and the parasympathetic. So let's write sympathetic here. And parasympathetic. So same spelling, parasympathetic. I've just shortened it there. Okay. So the somatic nervous system, connects to muscles. And we're going to talk about, you know, muscles and all of that stuff over here. So this whole right side of the mind map is going to be about muscles later on. But first of all, let's dive into the sympathetic and the parasympathetic a little bit. So actually what we can do is we can kind of make a little table here and talk about the sympathetic and the parasympathetic, Now, if you're using the textbook, then on page 92 there's a full table, but I think the key points we'll just jot down. So the sympathetic nervous system is basically about sort of speeding up. I think of it as speeding up, and the parasympathetic is slowing down. But more accurately, we could say that the sympathetic prepares for activity, prepares for physical activity, whereas the parasympathetic sort of prepares for rest. Let's do that in blue, sorry. So we can sometimes call it rest plus digest. Okay, so your body's either gearing up, ready to do some physical exercise, to run away from from something or something like that, or it's kind of getting ready for sleep and resting and digesting and all that kind of background sort of maintenance that needs to go on. Okay, what else can we say? The neurotransmitter. So NT for neurotransmitter equals noradrenaline. And on the other side, the neurotransmitter equals acetylcholine. What else? So this one is kind of to do with stress. This one is to do with relaxation or sleep. And this one increases heart rate, HR, increases ventilation of the lungs, and decreases digestion. Okay, also dilates pupils, which sort of open up the eyes, getting them ready for, ready to see and evade predation, I guess. That's kind of the fundamental reason we have this is just kind of fight or flight. It's kind of similar to the adrenaline response. And it reduces digestion. The other side, we've kind of got the opposite. So it brings down heart rate, brings down ventilation, constricts pupils. So constricts pupils, that's the opposite of dilate. And increases digestion. Now, two examples of the nerves that you need to know. Sympathetic nerve that controls the heart rate is called the accelerans nerve, is the one increases heart rate. And on the other side, we've got the vagus nerve, which decreases heart rate. And both of those two things work by modulating the activity of the SAN, the sinoatrial node, which you should remember from year 12, and they act to kind of speed up or slow down the heart rate. But let's now go from there into the brain structure, which is going to relate to the control of the heart as well. Okay, so here we are at the brain, okay? The brain is shown over here. Key coordinating organ of the central nervous system. So first of all, let's sort of just label this diagram and talk about all the different parts. All right, so let's start with the cerebrum. Now, the cerebrum is what you probably think of as your brain. It is the part that is responsible for the for conscious thought. As you're doing this mind map right now, I'm hoping that your cerebrum is pretty active and you're thinking about it deeply. Conscious thought and sort of higher thinking, Okay, we've also got the skull here, just not really part of the brain, it's just the protective casing around the skull. Down in the middle, we've got the hypothalamus. This area here is the hypothalamus. I think I'm just going to actually switch it around though, and I don't want to label it over here. I want to label it on this side for purposes that will become clearer later. So this is the hypothalamus, and I'll put that in red because it's kind of, I'm going to talk about that in a minute. Hypothalamus. And it works together quite closely with the pituitary gland, which is right here. So they kind of work together, which is why I want them both on that same side. We're going to talk about those in a minute. And then here we've got, what else have we got? This is the cerebellum. So very easy to confuse the word cerebrum with cerebellum. Cerebellum, I think it's actually Latin, and it stands for, it kind of means little brain, because it looks like a little mini brain. I'll see if I'm going to just drop in a picture actually of a brain. Here you go, boom. There's a brain, and maybe you can see the little brain there, the cerebellum as well. Okay, it's gone. All right, then we also have, in here we have the pons, which is this area, and then we have the medulla oblongata, which is kind of here, and actually I'm going to label this over here. We have the spinal cord, which is part of the central nervous system. Okay, so I'm just going to get rid of that line there, and I'm going to get rid of that. Okay, so why did I put the medulla oblongata over here? Well, because I want to talk about one of the main things that's in the medulla oblongata, which is the cardiovascular kind of control center. So, yeah, let's put that down here. So generally, the medulla oblongata, it has a role of controlling a lot of the autonomic responses. Responses, which is why I put it here. Okay, so it links to this, which is not conscious. So, for example, the main one that you need to know about is the Cardiovascular Center. And the Cardiovascular Center, how am I going to do this here? I'm kind of going to do a little bubble around it. I sometimes like doing little bubbles like this. Now, the cardiovascular center receives information from loads of different places, and it kind of decides what to do. Should it send a message down the accelerans nerve to speed up the heart, or should it send a message down the vagus nerve to slow down the heart? So, so in terms of things that will make it speed up the heart, we've got stretch receptors actually in the muscles. So when your muscles are moving, your medulla oblongata recognizes it and it speeds up your heart, because if you're moving your muscles, you're probably going to need more oxygen delivered to your muscles. Also, low blood pH is going to trigger the cardiovascular center to speed up the heart. Okay, so I've written there the thing that causes the cardiovascular center to trigger the heart to slow down would be high blood pressure in the carotid sinus. The carotid sinus is actually about here, and it's a region where various arteries come together, and those arteries basically have something called baroreceptors, which are pressure receptors, detecting the pressure of the blood that flows through them. So that cardiovascular center, remember, it works by then deciding whether to send messages via the accelerans nerve here or the vagus nerve here. Okay, so it either uses the sympathetic or the parasympathetic to control the heart rate. Okay, so that's a little bit on the brain. Let's keep going by looking now at the hypothalamus and the pituitary. I realise now that I haven't labelled one part of the brain over here on the left, which I think is the corpus callosum. That's right. Which is something that joins the two hemispheres of the brain. So let's just write that here, joins hemispheres. And actually the cerebellum, we need a little bit more information on there as well. Put this over here. So the cerebellum controls balance plus fine control of movements. And you could, I wouldn't use this in an exam, but the idea of muscle memory, that's not, muscles don't have a memory, but what muscle memory means is once you've practiced a movement loads and loads of times, you can kind of do it automatically and with finesse, smoothly. And the cerebellum is actually controlling those fine control of your movements. For example, when I'm writing, I don't need to think about the actual control of the pen as I'm writing. My higher thought centers in my cerebrum just tell my hand, write the word cerebrum, and then my cerebellum controls the kind of the fine control of my movements, if that makes sense. Okay, so the hypothalamus and pituitary, we're going to talk about those now. Now, the first thing about the pituitary and hypothalamus is the pituitary is a gland, which means it produces hormones. Okay, and it produces loads of different hormones. For example, we've got ADH, which we talked about when we looked at the kidney. We've got stuff like FSH and LH in control of the menstrual cycle, and a load of other hormones, for example, involved in the fight or flight response. So you could make a list of a couple of those, if you want FSH, LH, ADH, etc. But we're going to look a little bit in more detail about how these two together control the fight or flight response. Okay, and it's a bit complicated, so I'll try and keep it brief, but let's get into it. So hypo, for hypothalamus, it can release a couple different hormones. So it can release a hormone called CRH, which stands for corticotropin releasing hormone, which then causes the pituitary, so I'll just write pitu, which then causes the pituitary to release ACTH, which is what on earth does that stand for? Adrenocorticotropic hormone, which then actually goes through the blood and then goes to the adrenal cortex. And it causes them to release things like glucocorticoids, cortisol, corticosteroid hormones, so cortisol, etc. Okay, that's part of the fight or flight response. And the other part is that the hypothalamus can release something called TRH, which stands for, what is it called again? Thyrotropin releasing hormone, and that goes to pituitary and causes it to release something called TSH, which is thyroid stimulating hormone, and that then goes to the thyroid gland, which causes it to release the hormone thyroxine, which speeds up metabolism, okay? Up metabolism. But the other part of the fight or flight response is that the hypothalamus can also activate the sympathetic nervous system, all this thing over here, which then can trigger the release of adrenaline. So let's do that sort of over here. I mean, let's do that different color. So sympathetic nerves, and that will go all the way down to the adrenal medulla. And that releases adrenaline. Okay, so that covers pretty much all of the control of animal responses. So actually we started mainly with module sort of 5.5, which is animal and plant responses, but we've done all the animal stuff there. No, we've done all the control of animal stuff there. We're going to get on to muscles later on. But now I want to actually go back to neuronal communication for a minute, okay? Okay, so now let's zoom in and focus on the nervous system. So the nervous system, the brain, it's all made out of neurons, okay? So these up here are the different types of neurons that you need to know about. So neurons are nerve cells. And they all work via transmitting electrical impulses. Okay, and the way that they do that, the way that they transmit an electrical impulse is via something called an action. I'm trying to write sideways here, it's not going brilliantly. Action potential. Okay, so we're going to talk about the detail of that action potential using this diagram here in a minute, but not just yet. Now, for an electrical impulse to be transmitted, they start out in a polarized state. Okay, more on that later. So they have an electrical polarization of their membrane, and the action potential can transmit electrical impulse along their length. Let's zoom in a little bit more, and let's talk about these three different types of neuron. So, first of all, A, you need to recognize this. This is a motor neuron. B is a sensory neuron. And C is going to be a relay neuron. And now let's label these little bits and bobs. So this is the cell body. These are dendrites. This is the axon. This whole long purple thing here is the axon. And this here is called the axon hillock, which is kind of the start of the axon, where it connects to the cell body. The direction of transmission is this way, down here. But in this case, we've got a myelin sheath around it. I'm going to put that in red because that's kind of important. And actually you can label that over here as well. So the myelin sheath is this orange layer, and I'll also label it over here as well. So the myelin sheath can be on both motor neurons and sensory neurons. And the reason it's so important is that it causes transmission. So I'm going to just write increases the speed of transmission. So trans from transmission. And the key word you need to know there is saltatory conduction. I'm actually going to just scribble out this bit here so I can write underneath a bit more. So we'll talk about saltatory conduction a little bit more once we talked about the action potential and when we write a bit down there. But basically, saltatory conduction means that the AP action potential jumps. Saltatory actually means jumping from node to node. Node, you might say? What node? This node here. The little gaps in the myelin sheath is called a node of Ranvier. And because the action potential kind of doesn't, if it's myelinated, the action potential doesn't move smoothly along the axon, it jumps, 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 jumps. It means it goes faster. And the other thing about the myelin sheath that we need to know is it's created by a Schwann cell, so maybe that's, I'll put that over here. Made by Schwann cells. And myelin is a fatty material. It's a kind of lipidy-based substance, and it insulates the electricity essentially in the axon, basically. Okay, right. If we move on to the sensory neuron here, we've got the cell body. The direction of transmission in this case is this way, which means that over here we've got the dendron, cell body there, and then we've got the synaptic endings. So we said that the sensory, actually over here, we said that the sensory part of the nervous system carries impulses to the CNS, for example, the optic nerve. So we can see that here. The direction of the transmission is this way, and it's going, you know, to the CNS up here and from receptors down here. So, for example, receptors in the retina up to the CNS, whereas the motor neuron goes from the CNS up here. The direction of transmission is this way. And it could often connect to, might even connect to a muscle, or it might connect to another nerve first, and then eventually it might end up to a muscle, or also to a gland. And the relay neurons are generally ones that connect nerves to nerves, so they generally connect motor to sensory. Okay, so they're the ones that join up the motor and sensory neurons, or they might even, relay neurons might connect to other relay neurons. And actually most of the neurons in the brain I believe we class as relay neurons. Here we've got dendrites. This is that's not correct. That's kind of pointing towards the nucleus. The cell body is this whole thing. So that's the cell body, is big. And then these are the synaptic endings. And just a word on synaptic endings. The brain is so amazingly complex and clever because it has trillions and trillions and trillions of synapses. You know, each neuron in the brain might connect to maybe 1,000 other neurons with 1,000 synapses. So when you take the several trillion neurons in the brain and multiply that by several thousand, you've got, you know, lots and lots of synapses in the brain. Okay, so those are the different types of neuron. Just want to make a little side note, and I haven't really got space on it, but you do need to know about how these three types of neuron fit together in a reflex arc. So perhaps you could add a little bit extra up here on the reflex arc, and you need to know about cranial reflexes, which are really, really fast, and spinal reflexes, which are also very fast. And you need to know about the reflexes basically are fast and automatic. But you also need to know that some reflexes can be overridden. For example, if I touch my eyelash, you might notice I blink there automatically, but I can override it. If I concentrate and I wiggle my eyelash, it doesn't feel great, but I can override the reflex because the science behind that is because I send a fast message down a myelinated neuron, which overrides a slower message which is traveling on a non-myelinated neuron within the reflex arc. Okay? All right, let's now get into the really nitty-gritty of how the messages travel the action potential. Okay, so now we're going to talk about the action potential. It's going to be a little bit of a tight squeeze, so I'm going to have to kind of explain it, but then keep the notes a little bit brief here. So first of all, let's talk about what's going on at point 1. And also on the diagram below, let's just make sure that we're aware that this is the inside of the cell and this is the outside of the cell. And then actually I think what we'll also do is we will just use a bit of a color code for the position for the different ions actually. So I think I'm going to do red for sodium. So let's just make all these sodium ions red. It's going to be a bit helpful to have the sodiums and potassiums different colors. So yeah, we've got the, I'm actually going to go over the potassiums as well. I'm going to go over them in blue. So we can already see here there's a gradient of sodiums on the outside of the cell and potassiums on the inside of the cell. And that's a significant point. So at 1, we could say the neuron, because we're talking about neurons here, is polarized. And it's polarized to around about -60. So this is -60 here. And there's two things that are going on to cause that. First of all, the sodium-potassium pump, which takes 2 potassium ions and moves them into the cell. Sorry, 3 sodium ions and moves them out of the cell. And 2 potassium into the cell. So 2 potassiums go in, 3 sodiums come out. So there's a kind of a build-up of like a -1 on the inside of the cell because we've moved different amounts of positive ions. And the second thing actually is that there are K+ channels open, which means that even when those potassiums move into the cell, some of them actually leak back out. So I'll just kind of draw that here. So this potassium, let me just zoom in further, because we're going to have to get quite close up. This potassium here also can kind of come back out again, and I'll just write channel there, which helps build up this resting potential. Now, for an action potential to be triggered, there needs to be something happening, and this could be a synapse connecting to a neuron and causing a generator potential. Or it could be, if we're talking about a receptor, it could be the receptor itself starting to be depolarized due to receiving a stimulus, and we'll talk about that in a little bit. But before the action potential starts, let's look at the status of this here. This is a sodium channel, and it's closed. Now at point 2 here, some sodium channels open. This is actually a K plus channel. So at point 2, I'm going to alternate colors to kind of make it a bit more easy to follow. So at point 2, generator potential, some Na plus channels open. So therefore, the sodium ions start moving down their gradient. So actually this 2 here also refers to what's going on here. Yeah, this is kind of 3, 4. So this is a sodium channel open. So we have a generator potential. Some sodium ions start to move into the cell, and they're positive, remember? So as they move into the cell, they make it less and less negative. So we have a depolarization, moving from -60 to -50, up and up and up. Now, once they reach a threshold value, once the cell reaches a threshold value of about -50, which is about here, then we get the triggering of an action potential. And once it's got to that point, the action potential will definitely occur. Okay, so at 3, we reach threshold potential. Reached, which is -50 millivolts. And once that occurs, we trigger the opening of voltage-gated sodium ion channels. So that's what's occurring at 4, or sort of around. That's actually sort of around here, really, 3 or 4. So I'll put this down here. Voltage-gated sodium channels open. And once those channels open, more and more sodium ions flood in, which causes the membrane to depolarize even further. And actually goes so far that the inside of the membrane becomes briefly positive. So it kind of reaches a maximum of about +40 up here. I'll do that in blue. So actually at 5, membrane reaches +40 millivolts as sodium floods in, because there's a big concentration gradient. So loads of sodium floods in, and it takes it to the positive region. So how come that doesn't just keep going? Well, there's another ion channel that we need to consider, which is a potassium ion channel. So potassium ion channels then open up. These are voltage-gated potassium ion channels. So that's what's occurring here, right here. So there's kind of a switch around this point. So this is kind of point 6, which I will label down here. So here we have Na plus channel closes, which is this one, and K plus channel opens, which is this one. Okay, so the sodium channel, which is voltage gated, is really interesting kind of channel, and it actually has almost like a timing mechanism. So once it's been opened, remember the sodium channel kind of opened around about here when we got to threshold potential. Then it kind of has an inbuilt timing mechanism, and it shuts a few fractions of a second afterwards. In fact, we could look at the timeline here, and we could say that it's probably about 1 to 2 milliseconds later it will shut, and then the potassium channels open a little bit later on during the action potential. Once the potassium channels open, we then have potassium ions moving. So potassium ions, remember, they are a higher concentration inside the cell, but they flood outside. They moved out of the cell, out of the cell, out of the cell, taking the membrane potential back into the negative region. So let's just add a few more potassiums sort of moving out this way. We've got potassiums flowing out now. And actually, I'm going to add over here as well, just to show that when we're doing the action potential here, we've got sodiums flooding in. Let's add some sodiums right there, because that's what causes the... So we have sodiums flooding in here, causing the membrane to go positive, and then potassiums flooding out, causing the membrane to go negative. So that's what's occurring at 7. So 7 is basically repolarization. So 7, repolarization as K plus ions exit down concentration gradient. Remember, all these ion movements are down concentration gradients. The concentration gradient itself is set up at 1 here by the sodium-potassium pump, but then once it triggers, it's just diffusion, facilitated diffusion through these channels. Okay, now the potassium ions leave so quickly and so rapidly that they briefly take the membrane into a sort of state of hyperpolarization. So actually, you can see here, this is the normal kind of level of polarization about there, but at 8, it briefly overshoots. Maybe it goes to like minus 70-ish, and we call that hyperpolarization. And then finally, it returns to its resting state. Briefly, there is what's called a refractory period, where those voltage-gated sodium channels are kind of timed out. Remember I said they have a kind of timing mechanism in there. So after they've been opened, they kind of shut, and they will not open again for a couple milliseconds. So there's this refractory period where once a neuron has been sort of fired, and an action potential has been fired, it can't fire again. So at the end, let's just label these things here. So at the end, after the hyperpolarization, finally, at this point here, K plus channel closes. Remember, they were open, causing the repolarization. But the Na plus channel stays closed, because it closed over here, remember, and it's still closed over here. And it's the action of the sodium-potassium pump that brings things back to normal. So NAK pump restores resting potential. Okay, so that is the action potential there. Okay, I just want to go over that one more time using this animation, because it is a very tricky part of the syllabus, and I wanted to show you what's going on in the ion channels in a bit more detail visually. Okay, so here's a nervous impulse. Now we know that at resting potential, we have a difference in charge. The inside of the cell is negatively charged, and the outside of the cell is positively charged. So we say that it is polarized. And this is mainly due to sodium and potassium. There are way more sodium ions outside of the cell and quite a lot more potassium ions inside the cell. This gives a charge of about minus 70 millivolts. Actually, in your syllabus, I think it's minus 60, but sometimes it's minus 70. I think it may actually depend on the neuron. I have seen one exam question which asks you to sort of analyze some data and compare neurons, for example. So it can be 70 or 60. All right, so when the action potential starts, we use voltage-gated channels to kind of transmit it. So initially, there is a small depolarization. Some sodium ions move in to the cell. There they are, going down their concentration gradient, moving into the cell. This causes the inside of the cell to become depolarized. It reaches threshold potential and causes positive feedback. So as the inside becomes more positive, more and more sodium channels open, more ions move in. This is positive feedback. So how does the action potential spread? This is the really interesting bit which I wanted to show you actually. I'm going to take that back again. So as sodium ions move in, let's pause it there. As sodium ions move in this region of the cell, and it briefly becomes positive, like that, as the sodium ions move in, they don't just stay exactly in that part of the cell, do they? Because they're ions and they're free to diffuse. So as they move in, they turn that bit positive, but they might spread a little bit down the axon, turning the next bit of the axon positive as well, enough for its sodium channels to open. So it's the diffusion of ions just in that little local area that kind of causes the next set of voltage-gated sodium channels down the axon to open. So it spreads like a wave, propagating down the axon like this. But the action potential is an all or nothing response, and it can only move in one direction down the axon, okay? Because now this part of the axon here, as we talked about before, would be in its refractory period. So it can't be polarized again, and the action potential moves this way, sorry, depolarized again. So repolarization occurs when the sodium channels close, because they've got that kind of timing mechanism. This says 0.5 milliseconds. And now potassium channels open, allowing potassium to move rapidly out of the axon. This causes a brief hyperpolarization, which is shown here in green. And then eventually the resting potential is restored through the action of the sodium-potassium pump. Now, I did say at the beginning that we needed something to trigger the action potential, and that's just reminded me that we didn't quite put a lot of info down here, so we're going to. So the sensory branch of the nervous system, let's just put up here, connects to receptors. And receptor cells, I'm just going to kind of put a little box around here for receptors, okay? I'll just put a bit of notes in here. So receptors are electrical transducers. They're transducers, sorry, I'm not sure if I would say electrical. They're just transducers. So they're transducers, which means that they can take one stimulus and turn it into an electrical signal. So for example, rod and cone cells in your eyes take light, the signal, and transduce it into an electrical signal. So let's just write e.g. light to elec, okay? And they all contain gated ion channels. So for example, the cone cells and the rod cells in your eye, they have a pigment called rhodopsin, and when a photon of light hits that rhodopsin, it changes its shape, and that change of shape eventually leads to an opening of an ion channel, and sodium ions flood in, depolarizing the receptor cell. You have like hair cells in the ear. Actually, the sound wave kind of basically bends the cell in a certain way, and that bending opens a little channel with a trapdoor kind of mechanism. It's really fascinating how it works. So gated ion channels, gating ion channels, let's say channels open, plus sodium goes in. And this is a common mechanism. And the ones that you really need to know about, for example, is you need to know this one, Pacinian corpuscle detects pressure. And I think that's all you need to know about sensory receptors, really. Yeah. and we call these sensory receptors, actually. Let's call them, give them their full title, sensory receptors. Okay, all right. Okay, so we've looked at the action potential, but how does the action potential move from one neuron to the next at the synapse? That's the next thing we're going to look at down here in the bottom left-hand corner. So let's zoom in. I'm going to try and go through the synapse pretty quickly. I think you should be able to get it pretty well. Okay, so here is the action potential. Okay, action potential moves this way. So this whole thing here is the synapse. All right, so in the synapse, we've got mitochondria. Lots, okay? I haven't written that very well, have I? Lots of mitochondria. In the presynaptic bulb. So this whole thing here is called the presynaptic bulb. And it's got mitochondria. It also would have lots of endoplasmic reticulum because it's just quite a metabolically active place. There's a lot of transcription and translation going on to produce neurotransmitters and enzymes involved, and regulation of neurotransmitters and that sort of thing. So as that action potential moves in, it first triggers. So this is actually the first thing, number one. And then the second thing here is number two. So we have voltage gated calcium channels. So therefore, the calcium channels open, and we have a pre-existing concentration gradient. There's more calcium outside, so the calcium ions move inside, okay? Now that triggers point 3, which I'm probably gonna have to write underneath because I'm kind of running out of space. So one action potential moves in, 2 calcium channels open, point 3 vesicles. What's going on here? There we go. Vesicles containing neurotransmitter, neurotrans for short, move to this membrane plus fuse. So they release the neurotransmitter into the synaptic cleft by exocytosis, really. Now this is exocytosis that is regulated exocytosis. So it's regulated by the action of calcium. Calcium comes in and it allows the vesicle to actually fuse. Fascinating stuff on this. We won't go into it. Botox blocks this process. If you want to research that yourself, then please do. So zooming in a little bit more. So that's actually kind of 3 here. That's point 3, which I've written underneath. So now we have calcium released into the synaptic cleft. That's what this is called. This is the synaptic cleft. And in the synaptic cleft here are sodium channels. And these are not voltage gated sodium channels. They are gated by the neurotransmitter. So they are acetylcholine gated sodium channels. And we can write acetylcholine like this, ACH. Did I say the neurotransmitter? So yeah, neurotransmitter is acetylcholine. So these open, so point 4, once the acetylcholine binds, sodium channels open, Na plus in, and then you get depolarization, which triggers an action potential. Okay, One extra thing that you would need to know is that acetylcholine can be broken down by an enzyme. And this is a really long word, acetylcholine. I'm going to have to go into the next line, esterase. It's all one word, acetylcholine esterase. Otherwise, the kind of synapse would keep firing. And then eventually what happens is the acetyl part and the choline part, which have kind of been separated, they get reabsorbed back into the presynaptic bulb and joined back together, which needs ATP. So, so how do we draw that? Okay, so let's say we can do the acetyl part. Plus the choline part. This is me saying esterase breaks down to the acetyl part and the choline part, but they can be rejoined using, so it gets a bit weird down here when I'm trying to write at the bottom, it doesn't like it. Rejoined using ATP. So that's one of the reasons why we have lots of mitochondria up here, because it's a very energy sapping process. Also, of course, we've got lots of sodium potassium pump channel proteins there, which trigger, sorry, not trigger, which cause the resting potential. So we need lots of ATP. All right, so I said that depolarization results in an action potential, but does it always? Not necessarily, because what might need to happen is we might need to have some summation going on. So actually, what occurs, this depolarization leads to a tiny little blip, tiny little depolarization, but if the depolarization is not big enough and it doesn't reach the threshold potential, then there won't be an action potential in the next neuron. So what we can do sometimes is we can get something called summation. This is where the neuron after the synapse kind of adds up different stimuli to decide if it wants to fire. So in this case here, this is a diagram showing temporal summation. Okay, so we have one action potential, one pulse of neurotransmitters from the synapse and a bit of depolarization, a second and a third. So we have three action potentials coming into the presynaptic bulb and to the synapse in quick succession, like bang, bang, bang. And they build up. So the depolarization kind of builds up, 1, 2, 3, eventually getting to threshold and triggering an action potential. So then we would have AP triggered. Okay, so that's temporal summation. We can also have spatial summation. And that's when we have 3 separate axons all arriving at one synapse. And they all, if the action potentials arrive at the same time, they can all release neurotransmitters, triggering kind of 1, 2, 3 sort of depolarization in kind of 3 quick steps, almost actually at the same time, reaching threshold potential, and an AP is triggered. Not on the syllabus, but we can also have, actually it is briefly on the syllabus, we can also have summation of positive and negative inputs. So a neuron can be getting stimulated by one synapse, which causes depolarization. We call that an excitatory postsynaptic potential. So we need to label that. So like this here is called an EPSP. Excitatory postsynaptic potential. I haven't got the space to write it, but you should write it. Excitatory postsynaptic potential. So we can have summing of EPSPs through time, summing of EPSPs through space, and we can also have, this is kind of slightly off topic, but EPSP plus IPSP equals no fire. So if we have an excitatory postsynaptic potential and an inhibitory postsynaptic potential coming together at the same time, they cancel each other out. So some neurons can inhibit the firing of others. Okay, so we're in the home straight here. We're now going to get into muscles. So nerves can connect to things like glands. We said over here, where was it? Yeah, that motor neurons connect to muscles or glands. And of course, they can connect to other nerves. So we said that a synapse is where a nerve connects to another nerve. Now, over here, we have something which is very similar to a synapse, but it's called a neuromuscular junction. So that's where we're going to go next. So we could actually draw over here from this action potential, we could sort of go over here, connects to a muscle. If a nerve connects to a muscle, we have this, which is called the neuromuscular junction. And it's pretty much like a synapse. In fact, I'm not going to go over it in a huge amount of detail because it's so similar to a synapse, but I'll just label it. So if it is connecting to a muscle, then this type of neuron will be a motor neuron axon. And over here, this orange bit will be the myelin sheath. So most motor neurons and also sensory neurons are myelinated, which means that the action potential travels a lot faster. And actually, whilst I was refreshing my memory on this, I did find out how much faster. And actually, it's almost 100 times faster. So for a myelinated neuron, the speed of transmission it is about 100 to 120 meters per second. Let's just say 100 meters per second is the speed, whereas for an unmyelinated neuron, we're talking about sort of around about 10. So it's about 10 times faster. Okay, so back to the NMJ. So myelin sheath, motor neuron axon. Over here, we've got calcium moving in, so it's the same sort of thing that happens before. As the AP action potential arrives, we have voltage-gated calcium channels. They open up. Let me just zoom in a bit more. Calcium ions flood in. Then we have vesicles, which move to the membrane here and release the neurotransmitter via exocytosis. We have acetylcholine is the molecule in here. Now, one difference here is that we have to give this membrane of the muscle a special name. It's called the sarcolemma. And it's essentially, it's just the membrane of the muscle cell. The cell surface membrane of the muscle cell. Now, the same sort of thing happens in this membrane. We have receptors that the acetylcholine binds to, and then we have the movement in of sodium ions. So sodium ions move in, and then we have a wave of depolarization, an action potential. But in this case, the action potential spreads this way. Okay, it spreads along the muscle fiber. It spreads along the muscle fiber and actually down these things called a T-tubule. So this is different now. So here is a T-tubule. And this carries the action potential, the AP, into the muscle fiber. And to really kind of understand how this is working, we need to kind of look at the muscle fiber in 3 dimensions. So let's do that by looking at the diagram just below. So here we are. Okay, let's label this diagram here. So first of all, we've got the sarcolemma. So let's just join that here. This is the sarcolemma here, okay? The outer membrane around the muscle cell. Now, the T-tubules, which we've already talked about, that is here. Now, actually, T-tubule, T stands for transverse. Transverse means across. So these tubules cross through the muscle fiber. That's why they're called T-tubules. We do have a nucleus in here. But actually, what you might notice is there are multiple nuclei here of the cell, because, actually, it's not that clear here. But in a muscle fiber, individual muscle cells fuse together to form a multinucleated cell. So we have more than one nucleus in the fiber there. Also, inside here, we've got loads and loads of these. These are mitochondria. So this is a mitochondria here. And here, and here, and here, and here, and here, and here, all over the place, okay? Loads. Lots of ATP needed for muscle contraction. Now, if we zoom in even a little bit more, the T-tubules connect to something called the terminal cisternae of sarcoplasmic reticulum. Now, we've heard this word reticulum loads, like endoplasmic reticulum, smooth and rough. So it just means a system of membranes in the cell. So the sarcoplasmic reticulum is a system of membranes within the cell. And the T-tubules connect to that sarcoplasmic reticulum. What else do we need to look at? Okay, so how does this trigger muscle contraction? When the AP moves down here, how does it trigger contraction? Well, the AP, the action potential, arrives down the T-tubule and gets to the terminal cisternae. Okay, so forgive me, I'm going to have to cross over a line here. So, when the AP reaches the sarcoplasmic reticulum, there's a special setup which we don't need to know too much about, but there's these special channels that open, again they're voltage gated, and they release calcium from the sarcoplasmic reticulum into the sort of main body of the muscle cell. So AP reaches the sarcoplasmic reticulum, calcium released into muscle fiber. Okay, and that is what causes contraction. Contraction, and we're going to talk about that a little bit later, but it binds to troponin. Okay, so how does contraction actually occur? Well, first of all, it occurs by filaments sliding over each other. A little bit like my fingers here can slide to shorten or sort of slide out to lengthen. So the muscle fibers kind of do this. Okay, so I want to just take you through a kind of animation of this just to kind of help before we kind of get labeling. So muscle, first of all, there are three different types of muscle. We've got striated or skeletal muscle, we've got cardiac muscle, and we've got smooth muscle. We haven't really got space to go over that in this mind map, but potentially you could add it somewhere. Now, skeletal muscle, also called striated muscle, is striated because striated means stripy. Now, a muscle consists of many bundles of fibers. So here's a bundle, and if we go into it, we can see that there is one muscle fiber, and it is a long cell, which is, like I said before, contains many nuclei, so it's multinucleated because cells are kind of fused. The membrane around the outside is called the sarcolemma. And if we look within one muscle cell, we can find one individual myofibril. So each muscle cell contains many myofibrils contained within the cytoplasm of the cell, which is called the sarcoplasm. So sarcolemma is like the membrane, and then sarcoplasm is the stuff in the inside. Now, when we look at the myofibril, it looks stripy. And the stripes are caused by regular repeating sarcomeres. Okay, a sarcomere is kind of one band, basically, and it's the functional unit of the muscle. So each sarcomere can kind of contract a tiny bit and lengthen a tiny bit. It can generate a force contracting. It can't generate a force lengthening. It has to be stretched back out by a muscle acting in an antagonistic way. And within the sarcomere, what's actually going on is protein filaments are sliding past each other. Okay, the proteins can slide and shorten, and then when they're relaxed, they can be stretched out again. So, if we now look at the kind of history of the discovery of this, when scientists first looked at the muscle with an electron microscope, they saw an image like this. And this image, is stripy. So this is why they called it skeletal muscle or striated muscle, because they saw these stripes. And then they started to investigate what caused the stripes, and that's what led them to identify these sarcomeres. So the sarcomere was defined as the distance between these two dark lines, between the Z lines. Okay, so now let's look at this animation here, and you'll notice that this is on your mind map, so you can go ahead and label the mind map using these terms. Okay, so the sarcomere is the basic unit from Z line to Z line. So the Z line is the limit of the sarcomere, so you could label the Z lines there, and they appear as dark lines under the microscope. In the middle, we've got a different line called the M line. And the M line is found at the center of the sarcomere. And it's at the center, it attaches to the myosin filaments. So we haven't labeled those yet, so let's do that. So this thing here are myosin thick filaments. Okay, so this is the myosin thick filament. Now, in the center, the filament does not have any myosin heads, but towards the outside, it has these myosin head regions that can bind with another filament, which is called the actin filament. If you need to pause, obviously, pause the video and label this. But now I'm going to go on to the actin filament. So the actin filaments are also known as thin filaments, and they are formed from small protein subunits arranged in a helical structure. There's actually two kind of actin filaments, and they kind of wrap around like that to form a kind of helix. We've also got these bands here. So, and the bands really just refer to the different color bits of the stripy sarcomere that the scientists originally saw. So the I band is the region that has only got actin filaments, where there is no myosin kind of in. So if you look at my hands, I guess, the I band is kind of like the bit where my fingers don't overlap. And then you've got this band here, the H zone, which is within the A band. So that's, let's go back actually. Let's do the A band first. The A band is the part where the thick filaments are, so the part where there's myosin. But within that A band, which is large, there's a smaller sub part of it, which is called the H zone, which is where there is an actin. Okay, again, pause if you want to. I'm going to move on to the H zone now. There's the H zone. And then we've got the I band on the other side over there. Okay, so pause if you need to and finish labeling that. Okay, so now let's look at how contraction actually works. The sliding filament theory. So we're gonna look at how the sarcomere contracts. So here's the kind of filament, and what the scientists noticed when they were looking at these filaments under the electron microscope is that they move backwards and forwards like this. They saw this regular kind of change of colors. Okay, so we can see there that when the sarcomere contracts, three things happen. When the sarcomere contracts, what's happening to the sizes of the I, the A, and the H band? So when it contracts, the I band decreases in size. The I band decreases in size. The H band in the middle basically disappears. And the A band stays the same size. Okay. Let's jump out and go and label this again, actually, because we've covered a lot already. So we've got I band, A band, H zone, we've got I band, we've got Z line, we've got M line, we've got actin is the thin filament, and we've got this one here is the myosin, which is a thick filament. And then also we need to know about what's happening during contraction. So decreases in contraction. So does the I band. The I band also decreases during contraction, whereas the A band does not. It's the same size, because we've got the sliding like that. Okay, let's go back to that animation. So how is this actually occurring? So it's occurring because there's an interlocking between the myosin heads and the actin. So what they do, the myosin heads, is they bind to the actin. We call that a cross bridge. So they bind, they form a cross bridge, and then they can actually kind of pull and drag the fibers across each other. We call this pulling the power stroke. So let me show you that again. The myosin heads bind, form a cross bridge, and then they do a power stroke to just ratchet the fibers across each other. The myosin heads then unbind, and then they use ATP to kind of go back to their original position where they can do another power stroke. So the ATP is required to unbind them, and then they can power stroke, ATP unbinds them, power stroke, ATP unbinds them, and so on. When the muscle relaxes, what's actually occurring is the myosin heads detach, and then another muscle can pull the fibers out again. Okay, so let's just go back to the mind map here because I realize I want to add a little bit extra here. So we kind of talked about muscles, okay? So this is up here, muscle fiber. This over here. And over here is called the sarcomere, which is the functional unit of the muscle fiber. And it gets shorter in contraction. And over here, we need to just label these things here. This is the Z line. This here is the, well, that's actually pointing to the M line. The H zone is the white bit. So M line and the H zone around it. And then this distance here is the A band, because we can see that's where the thick filaments are. That's another Z. And this white bit here is the I band. So the sarcomere is from Z to Z. So I guess we could sort of mark that on sort of here. From Z to Z is the sarcomere. Okay, so now let's talk about a bit more of the molecular detail, and it's the last thing I'm going to do, of how the cross-bridge cycle works. So that's this, cross-bridge cycle. And before we get into that, I actually want to just focus up here. So we said that the contraction, the calcium ions bind to troponin, and then the troponin, once it's bound to calcium, it moves tropomyosin out of the way. Which then leads to the cross-bridge cycle. Okay, so we're kind of going to go from there down to there. So the contraction, now we're going to look at how the cross-bridge cycle actually works. Going to go to an animation quickly before we then label this diagram. And that's the final thing, so stay with me. Okay, here's the cross-bridge cycle. So, just like I was saying before, in a relaxed muscle, troponin and tropomyosin block access so that the myosin head can't bind to the actin filament, okay? But when calcium is released from the sarcoplasmic reticulum, calcium comes along and it binds to troponin. There it goes, it binds to troponin, and the troponin then undergoes a conformational change and it causes tropomyosin to kind of move out of the way, exposing binding sites on the actin filament. So now you can see these binding sites are free, and this is where the myosin head will bind. So the myosin head binds. Okay, once it binds, then there is sort of some tension in the muscle, and after it binds, it undergoes a power stroke. Okay, you can see it there. Let me show you that one more time. So notice that the myosin head has already broken down ATP. So we've already got ADP and PI there. So it binds to the binding site, and then it does the power stroke, and it moves along like this. Now ATP is then needed to unbind it. So ATP can now float in and attach, and that causes the myosin head to unbind, and it kind of like cocks, if you like, and it's now ready to kind of power stroke again. So it will then bind again, pull itself along again, and we get the cycle repeating. ATP causes it to unbind, power stroke, and so on, and so on, and so on. Now, an interesting feature of this is if you remove ATP from the muscle, then the fibers stay interlocked. Actually, I'll go back and show you that. So if you remove ATP from the muscle, so this ATP, let's say it's not there, if someone is dead, then the fibers stay interlocked, and that causes rigor mortis because they can't unlock without a supply of ATP. So how does a muscle stop contracting then? Well, the muscle stops contracting when calcium is removed from the muscle. So I'll just show you this a little bit again. When calcium is removed from the muscle and it's pumped back into the sarcoplasmic reticulum, then the muscle stops contracting and relaxes. And it does this when it's no longer receiving messages from nerves to keep firing. Okay, so let's see that here. Calcium is actively removed from the sarcoplasm when the muscle is no longer stimulated by a nerve, and then it returns to its original shape, and then it relaxes. So first of all, at point 1, myosin attaches. To thin filament. And then at point 2, we get the power stroke. And during that power stroke, ADP and PI are released from the head, from the myosin head. It's at stage 3, ATP binding allows the head to detach. And then it will actually undergoes hydrolysis. ATP is hydrolyzed. And that kind of allows the head to kind of spring back to its ready position so that it can bind again. So it binds again at 4. Let's just say the myosin head is back to the original position. And this cross-bridge cycle is occurring many, I don't know, thousands and thousands of times a second to ratchet those muscle fibers along. Right, we have made it, okay? We're there, we've done. That is everything. Everything about nerves, everything about muscles, and everything about animal responses. So we've actually covered over one module, sort of about a module and a half, which is why this video is so long. I'm sorry about that. So if you need to go over any more of that, and if you need to add any extra detail, we didn't quite get to talk about the 3 different types of muscle, smooth, cardiac, and striated, then use your notes to add more detail. All right, thanks very much, and I'll see you next time. Bye-bye.